All right, so we're looking at a cell membrane with a phospholipid bilayer, helps to prevent what can and cannot cross cell membranes. We know that things like ions, sodium, potassium, fluoride, calcium, cannot easily cross the cell membrane because they're polar. Keep in mind that polar, or ions, which are always polar, cannot cross cell membranes by themselves. They need a channel of some kind, whether it's a leaky channel or a gated channel. With that, keep in mind that the big unique difference between leaky channels and gated channels. Leaky channels require a stimulus. Gated channels do. It means we can control if they're open or closed. Now, in terms of knowing which way something's going to diffuse across the cell membrane, there are various concepts that I want you to really focus on and work on between now and your final, because it's going to be a repetitive theme for respiration, for the renal system, for digestion, when you get into those topics in 242. And that is what determines or influences something's ability to diffuse across a cell's membrane. We have two different types of gradients to think about, right? A concentration gradient. Things always diffuse from regions of higher to lower. Then ask yourself, well, that thing that's trying to diffuse, is it polar? Yeah, then it needs a channel of some kind. But also the electrical gradient has an influence on something's ability to diffuse across the cell membrane. When positive things move into a cell, it tends to make them more positive. When negative things move into a cell, it should make them more negative. When positive things leave a cell, it should make them more negative. So we're going to be looking at that a little bit more in tonight's lecture. So besides the chemical gradient and the electrical gradient, think about the types of channels, whether they're gated or leaky, and also transporters. And transporters have to physically, literally bind to something and move them across the cell membrane, like we have up here for a glucose transporter. But they work only in the direction of the concentration gradient. So they're always moving from high to low. In this case, the region from high to low is from outside to inside. But if you could make the cell have high glucose inside, this very same structure will then facilitate the diffusion from inside to outside. We have a term that talks about if it's moving two things in the same direction. You guys remember what that term would be? Moving two things in the same direction? Symporter, awesome. How about David then the other one? Importer. Which is two things in different directions. Awesome. Okay. You must have a lot of espresso. Okay. Now, keep in mind then that if you move against the concentration gradient, if energy has to be applied, we're looking at an active transport process. Now, the one you have to memorize right now is that you have a sodium-potassium ATPase pump, which is moving 3 sodiums out, 2 potassiums in. You want to maintain the chemical concentration gradient. If you anchor to the idea that any time an ion crosses a cell's membrane, that ion must be put back where it came from, then it's a no default thing to think about, that if I'm moving calcium ions, I need a calcium pump. If I'm moving sodium potassium, I need a sodium potassium pump to move them. Then we had this other bizarre one known as a secondary active transporter. There are lots of different kinds. So that's the general classification for it, a secondary active transporter. Specifically, this one is known as a sodium glucose co-transporter, where you're moving glucose against its concentration gradient. Normally you would think that if you're moving glucose from regions of lower to regions of higher, that this must need ATP, needs energy. Well, you're right, it does, but it's actually the energy that we would get in allowing sodium to follow its concentration from regions of high to regions of low. That's where you get energy from. And we're maintaining that ability because it's usually associated with your friend, the sodium-potassium ATPase pump, where we're constantly maintaining high amounts of sodium outside. So I have a facilitated diffusion of sodium inside, and that allows me, or this cell, to transport glucose against its concentration gradient. So keep in mind then that things that will influence a substance's ability to diffuse across the membrane is the concentration gradient, the electrical gradient, if applicable, the type of channel, the numbers of channels, and then the type of transporter and number of transporters. Right? That is so influential that those of you that just had lab, that's one of your questions in that lab sheet to drive that point home. Those of you that have not yet had lab, you'll see that question in the worksheets. Those ideas right there about what determines or influences diffusion across cell membrane is a repeating theme for many different systems in our body. So make sure you drum that into your memory, and we'll pound you a little time I've got left with that. All right, now the last thing is look at the idea of...
Can you expand? It's on the... You look at your computer screen over here. Is it... Can you make the slide bigger like it normally does? Just so...
You make me... Okay, is that better?
Yes.
Okay, there you go. Okay. All right. Well, I only had a few slides left to go. All right. So, those of you that just had lab, we've already talked about this, but we're going to drum it into you and give you a real-world application to this as well. All right, so now we're still looking at diffusion, but now it is the diffusion of water. Therefore, you have to change the terminology. We're looking at the diffusion of water across a semi-permeable membrane. That is what osmosis is. It's diffusion of water. And you must incorporate into your answer for the full 2 points across a cell's membrane. Otherwise, it's simply just boring diffusion. It's not osmosis. Now, if diffusion is across a cell's membrane, we need to know where there's high amounts of water and where there's low amounts of water. And the Yahoo that created this terminology decided to focus on what are known as the solutes, the dissolved particles, rather than direct up on water itself directly. So, we have 3 terms to keep in mind. One is hypertonic, right? High concentration of solute. That's the stuff dissolved in the liquid. If you have high amounts of solute, then you just simply learn that the opposite is true for the amount of water. If you're high in solute, you're low in water. Correspondingly true, then if you have a hypotonic, where by definition you are low concentration of solute, then the reverse is true for the amount of water you have. You have lots of water, but relatively low amounts of solute. These words are used to describe 2 environments that are adjacent to each other. So if one environment is hypertonic, and we're going to see in a moment, then the other environment is going to be hypo. They're always going to be opposite of each other. That will let you know which way water will osmotically move from one region to another. Water always goes from regions of higher to regions of lower. The easy way to think of it is that water will always diffuse or osmotically move into the more hypertonic environment, trying to dilute the number of particles to raise the amount of water, because that's the area with less water in it. Let's look at it in a real-world scenario, in a sense. So I've got 3 different environments, or a few different experiences. There's a beaker, that's a hypotonic solution. That's the beaker. That means that the beaker water has low amounts of solute, but high amounts of water. And because the beaker solution is hypo, that means the environment in the cell is hyper. It's high solute, low water. And if osmosis is the movement of water from regions of high concentrations of water to regions of low concentrations of water, the water will diffuse into the cell, trying to dilute that hypertonic environment. And the cell will expand. Now, if you're over here at the far other end, if the solution the red blood cell is in is a hypertonic solution, that means the solution outside the cell has high solute, which means low amounts of water. That means automatically the cell is hypotonic, which means it has low solute but high amounts of water. And now we know where our high and low water values are. Since there's more water in the cell, water will diffuse out of the cell, trying to dilute the outside environment. Okay? So water always tries to diffuse to the more hypertonic environment. There was to put this in a real world scenario, there was a game contest many years ago called Hold Your Wii for a Wii. It was a drinking game. When the game console Wii came out, a radio station, I think it was in Denver, I think, said, hey, we got a brilliant idea, let's have a drinking contest and we're going to see who pees last. The person that pees last wins a Wii console game. So hold your Wii for a Wii. Sounds brilliant. They had people drink 125 milliliters about every 20 minutes. So one of these every 20 minutes. How many of these can you drink before you have to pee? Well, gal won. She was the last one to have to go pee. Drank about a gallon and a half of water overall. She was excited. Cool. She did a no-no. She texted while she drove. Told her husband, honey, I won. That's fantastic. Hey, I got a bit of a headache, but no big deal. Next morning, she was dead. All because of this. Now, think about this. So, crash course in digestion. You drink water, it does not absorb from your stomach. It's going to go into your small intestine. And you're going to absorb the water from your small intestine. It's going to go into your blood supply, into your bloodstream. So normally, you guys are isotonic, right? The concentration of solutes in the cell is the same as it is outside the cell. Now you start drinking. You start adding more water to the blood. More water to the outside of the red blood cell. Does that make the blood hypo or hypertonic? You add more water to the outside environment of the red blood cell. You're going to make it hypotonic. That means the plasma has a higher region of water than, relatively speaking, inside the red blood cell. Water was diffused into her red blood cells. You can take a little extra water because it's a plasma membrane. It can swell a little bit, but not a lot. She was having what's called hemolysis, red blood cell destruction. Her red blood cells were popping left and right. Now, normally you do that anyway. You lose about 3 million a second. But she was losing a lot more than that. Plus the increase in blood volume. She was rupturing cerebral capillaries. She was bleeding internally, and there's no way to stop that. Once you have cerebral arteries in the brain rupturing, not a damn thing we can do for you just yet, other than try and put the blood in faster than it's leaking out. So her brain swelled up, her capillaries were rupturing, and her red blood cells were rupturing. And she died the very next morning. All because of drinking water. Was there an easy solution to this? What's the easy solution? Drink an isotonic solution, right? So you're 0.85% sodium. Drink a 0.85% salty water solution. Or do something that's a little safer, like Gatorade, Powerade, that have electrolytes in it. You know, won't be nearly as dangerous as this. And you'll get sick of the flavor before you have to pee anyways.
So if she were to, at that point, was there anything she could have done? If she
I think her name was on the tombstone the minute she started having severe headaches. I don't think there's a thing they could have done for her.
Or even after urination, it wouldn't have removed enough?
Well, the only possible chance would be, that I'll teach you guys if you have me for 242, is using what's called a high ceiling diuretic that affects the A-side and try and make her pee about 3 times every 20 minutes, way more than normal. So I'm going to make her produce a high volume of clear urine. At the same time, I'm going to need to replace her dead blood cells. So give her a red blood cell transfusion, not a plasma transfusion, and you might have a chance, but it would be a guarded prognosis. And depending on how bad it is. You know, if she came to me with I'm getting a headache. Oh, okay, what'd you do today? Drink no coffee? No, I had a drinking contest. What'd you drink? Lots of water. And if I was on my toes, I go, oh shit. Sorry. And start making you pee really quickly and already realize what's going to happen to you in 3 or 4 hours from now, you might have a chance. But if they just had followed a couple of nurses who called in and said, you better have her drink an isotonic solution, otherwise you're going to have a problem on your hands. Long story short, the radio station got sued for $45 million, got shut down, the manager lost his job, radio station was sold because they killed a mother and a wife from a very apparently innocent drinking game, right? So you can be killed by drinking just straight water. You drank too much of it in a too short period of time.
I used to backpack a lot in the Grand Canyon, and it wasn't uncommon to see people the same exact thing happen to them.
Yeah.
They lose their electrolytes, but like keep like hydrating a lot.
That's because you're sweating a lot.
Yeah.
You lose a lot of electrolytes. Yeah. It's unfortunate. That's why I bring salt and drink lots of water when I go. All right, last big idea is just to move large amounts of stuff, like maybe releasing neurotransmitters from a synaptic knob of a motor end plate region. Right? You have exocytosis. That's because the vesicle is made of the same stuff as your cell membrane. They are both phospholipid structures. So it literally merges, becomes a part of the membrane, and releases its content into the extracellular environment. You can do the reverse, called endocytosis, right, where you bring in substances, whether toxins or pathogens or nutrients, but you actually pinch off a part of the cell's membrane to create a vesicle, and maybe endosomes and lysosomes could come in, or you can break it apart and get to the glucose that could be inside of it. But endo simply means to bring in, exo means to release to the outside environment. And that's the last bit of those notes.
Can you just go over again, so it was the concentration gradient, the electrical gradient, and then it was the type of channel, the number of channels, and then what were the other two?
Well, you've got, those are the two on the slide. It was the idea of the type of,
Oh, the type of channel.
number of channels, number of transporters, so it's slide 5.
Okay.
All right, so now on to the next one, if I can get out of my notes here. And I'll make it full screen, so it's as big as possible. All right, you should know this slide by now. You should have your anatomy down. If not, it makes a nice little reference to all the various parts of a neuron structure. But I want to emphasize to you some very specific anatomical things about a neuron's membrane. And that is, if you look at this little flow chart, let me kill the lights for a moment, it's easier to see, that there are very specific types of channels found in specific areas of a neuron. If you look at the far left-hand side, this kind of yellowish beige color, that means that the entire neuron will have the following types of channels. It'll have sodium potassium pumps, it'll contain sodium leach channels, and potassium leach channels. Those are found everywhere. That means dendrites, soma, and the nodes, the node region especially. Don't forget the nodes. They will have those types of channels. But just the dendrites and soma will have chemical-gated sodiums, chemical-gated potassiums, and chemical-gated chlorides. They're only found on the soma and dendrites. That's it. If you look right in here at the initial segment, we have a crap load of voltage-gated sodiums. Your highest amount are found right here in the initial segment. And then lots of voltage-gated potassiums. And then for the rest of the axon, only exposed in the node region, are we going to have voltage-gated sodiums and voltage-gated potassiums. But don't forget, we also have these guys, right? This is now overlapping. They are saying that from dendrite to axon terminals, we have all these channels. But in these specific portions of a neuron structure, we have different specific types of gated channels. And then you get all the way down here to the axon terminal where we have a unique set also, and that is voltage-gated calcium channels. And don't forget, anytime an ion crosses the cell membrane, we need a pump to put it back where it comes from. Keep in mind, we have high sodium and high calcium outside, and high chlorine. We have low potassium inside. You want to memorize this because it'll help you keep track of what's happening where. Go to this part. Wow, we lost some light. All right. Remember that a membrane potential was the difference in charge across a cell's membrane. Keep in mind there are two different ways to think about membrane potentials. All cells will have a membrane potential. They'll have some kind of a difference in charge. All different types of cells will have one. But only muscle membranes and neuron membranes are excitable. So all cells have a membrane potential, but only muscle and neurons are excitable. That means they can experience a rapid change in their membrane potential. That means they can create an action potential, which is the same thing as a rapid change in the membrane potential. Keep in mind also that what's responsible for this charge across the cell membrane is the concentration of ions found inside and outside. That's what's creating that charge. If we change the concentration of ions, we are going to change the value of the membrane potential. And that's tonight's goal, to walk you through an action potential formation. All right, so think about what then an action potential is going to be. It's going to be defined as a brief transient change. Brief means milliseconds, like 3 milliseconds worth. Transient means it's moving along. So it's a very brief, rapid change in the membrane potential that moves along the length of our membrane. We need to apply a stimulus, maybe a neurotransmitter or maybe voltage, to actually change the permeability of a membrane. Remember, a membrane is semi-permeable. It is impermeable to ions unless you open up a gated channel. So we need a stimulus to open up those gated channels. We've got chemical gated and voltage gated. So we need to apply either a chemical or a voltage to make these gated channels open up. And that will increase the permeability. And then ions can move across the membrane. Let's see how we would do this. All right, so on the very top is just to remind you that a neuron's membrane has a resting membrane potential of negative 70 millivolts along its entire length. Whether you are in the dendrites, the soma, or along the axon, right, and you don't screw with it, it's mind its own business, that gives us a resting membrane. But keep in mind the various different types of channels, right? Here you have your chemical-gated potassium, chemical-gated sodium, chemical-gated chlorides. But along the axon, you have voltage-gated channels. Now we're at negative 70 on the inside. That's because you guys have way more potassium on the inside and lots of negative charged proteins. And there are more negative things in than there are out. You have potassium leak channels, so potassium is always leaking out. You have sodium leak channels, where sodium is always leaking in. I guess there's a sodium leak channel. And as long as you have this nice harmony of potassium leaking out and sodium leaking in, and you have your pump running at like a certain dance ratio, you're going to hover at negative 70. You guys have way more potassium leak channels than you do sodium. So with all that negative charge inside, as long as the amount of potassium leaking out and the amount of sodium leaking in is being held at a constant, and your pumps are running at a constant, it's like your engine idling at a stoplight, right? The RPMs stay at 2,000 RPMs. Now, if you apply the gas pedal, you increase the RPMs. If I apply a stimulus and cause either sodium or potassium channels open up, I'm going to change my resting membrane potential. Let's see how we do that. So let's look at this guy. We're going to go to the very end. We're going to go right here and remind ourselves what happens in the synaptic knob to create chemicals to land on the post-synaptic membrane. So here's my really big, huge, honking synaptic knob. We know we have a membrane that contains voltage-gated calcium channels. We have vesicles inside that are storing my neurotransmitter. I need to get those vesicles to fuse with the membrane to do exocytosis. So we have to have voltage, otherwise known as an action potential, come down that membrane and cause the membrane potential here to become more positive, so I can open up these voltage-gated calcium channels. And if we do that, I'm over here on the far right-hand side, then if I can open up these voltage-gated calcium channels, calcium comes in. let's practice an exam question. If I were to ask you, why does calcium come into the synaptic knob, what would you guys say? No. Good answer, but no. All right? Let me ask you this. Is it possible for me to open up these voltage-gated calcium channels and not have calcium come in?
Yes.
How so?
Concentration or electrical gradient are basically pushing them in, driving them in.
Okay, what if I did this? You're right. What if I removed all the calcium from the outside solution? I apply a voltage, right? Won't the gates open up? Yeah. Is calcium going to come in? No. Why not? Because there is no gradient. That's the why. All right? Yes, you gave it an ability to cross, but why was it actually diffusing into the cell? Because you have the concentration gradient where there's high calcium outside and low calcium in. What Brandon was referring to is, what if I created a scenario where I had high concentration inside compared to the outside, and then we applied voltage and opened up the voltage-gated calcium channels? Calcium will then diffuse out of the cell. All right? So the reason it's diffusing is because of the concentration gradient. That's why it moves. And I want to beat that into your brains. It's going to be a trivial, you guys know what Trivial Pursuit is? Or am I getting old now? All right? It is your trivia default answer to why things happen in the body because of diffusion. Things going from regions of higher to regions of lower. They may need an ability, but that's why they're moving, because they're going from high to low. All right, so the voltage comes down the membrane of my synaptic knob, opens up voltage-gated calcium channels. Calcium diffuses into the cell because there's more calcium outside than in. Calcium, binds to the vesicle. That allows for exocytosis of my neurotransmitter. Now I've got a post-synaptic membrane, and it has channels in the membrane. I can see that these molecules of neurotransmitter have landed on that binding site. This one has yet to get one. So this one's open, but that one's going to be closed. And these two are going to be open. So that means then whatever ions that are in high concentration out here, if they have a channel that's built for them, they'll be able to diffuse into the cell. Keep in mind that outside of your neurons, you have high calcium, high chloride, and high sodium. So if these are chemical-gated sodium channels, who's going to diffuse in? Who's not going to diffuse in? The calcium and the chlorides. Who's not going to diffuse out? The potassium, because you haven't opened up the gate for them. Yes, they have leaky channels, but they're always open. Right? We are applying a stimulus called a neurotransmitter. We are now opening up gates that were normally closed. So sodium is going to be able to rush in. So here's another way of looking at it. There's my cell membrane. This is the postsynaptic membrane. There's my neurotransmitter having opened up this chemical-gated sodium channel, and then sodium diffuses in to create a large cloud, relatively speaking, of sodium. And we've already sort of talked about this for muscles. So let's make this my chemical-gated sodium channel. Here's the neurotransmitter landing on it. I diffuse in as a big cloud. When you think about this, it's not just one sodium ion. It's thousands, maybe about 10,000 within a blink of an eye, have gone through that one little channel. What if you have hundreds of channels? That's a crap load of sodium diffusing into the postsynaptic membrane. I am now this big cloud of sodium. Where am I going to go? To wherever there's less of me, right? So if there's less of me in that direction, I'm going to diffuse this way. But now, we're inside of a neuron now. This is no longer a skeletal muscle. Think about where we are, relatively speaking. I'm diffusing over here, right, with my chemical-gated sodium. I need to get to here to start an action potential. So if I diffuse in from right here, and I diffuse this way, that's just totally useless. I need to diffuse as a cloud to the initial segment. So if we're looking at sodium diffusing in, as it moves both towards the initial segment, it can also then move up, it can also move away from the initial segment. So this isn't helping me. But as I move toward the initial segment, the amount of me is getting less and less. Right? This is what's called a graded potential. There is a weakening or a decrease in this sodium stimulus. That as it diffuses from its point of entrance, its effect gets less and less. I'm getting weaker and weaker the further I travel from where I came in. My ability to change the membrane gets less and less. There has to be enough of me to survive diffusing. So when I get to the initial segment, there's enough of me here still to make the membrane go from -70 to at least a -54. So I can pass threshold value and make these voltage-gated sodium channels open up. Remember, I came in through a chemical-gated sodium. So we open up chemicals with neurotransmitters, sodium diffuses in. It diffuses all different directions. Those that diffuse this way will hopefully reach the initial segment, make the membrane go from -70 to at least -55. Then we open up the voltage-gated sodium channels and we start all over again. But this ability of getting less and less the further I diffuse is called a graded potential. Graded potentials get weaker the further they travel from the point of origin. So let's sort of try something like this. Give you guys a couple of seconds to write those last bit of notes down. And have all of you pay attention for the next 20, 30 seconds. So let's see if this works. I think you heard what I said. All right. So, what did you hear, Brandon? Mumble, mumble, jumbo, right? The back row, who I was actually talking about, heard nothing of what I said. Why not? What was happening to the strength of my voice as it traveled further and further away from me? It got weaker and weaker, right? That's what's happening to sodium, right? Its effect is getting weaker and weaker from its point of origin. That's the whole point of like tossing a rock into a pond. As the ripples get weaker and weaker, the further they travel from their point of origins. Now, we have to have enough sodium come in to reach the initial segment. So this is why, in a sense, evolution, from my perspective, intelligent design, maybe for some of you, the synaptic knob releases well than more than enough, a crap load of neurotransmitters to get a ton of chemical-gated sodium channels open. It's overkill. I want to make sure I've got enough sodium to reach here to trigger the initial segment. We're not going to screw around. So we're going to give you way more than you need. So the membrane could probably bounce to a +20. But we're making dang sure we've got enough to reach the initial segment. Now, that means that we're going to open up a bunch of voltage-gated sodium channels. Which way is sodium going to diffuse then if we open up a bunch of voltage-gated sodium channels at the initial segment? All right, it's going to diffuse in. Now, why is sodium going to diffuse in at the initial segment? No. No. What is going to be your default answer for why things happen in the body? Concentration, but also now, what's the membrane potential before those sodium channels opened up? It was maybe a -55, right? Or a -50 maybe. It's a negative environment. What charge does sodium have? Positive. And where's there more sodium, inside or outside? So I've got two gradients encouraging sodium to diffuse in. It's the electrochemical gradient. That's why sodium comes in. It's very negative on the inside, and there's high sodium outside compared to the low inside. That's why sodium is going to be rushing in.
So is that called repolarization?
No, not at all. That's called depolarization, because you're depolarizing the membrane. We're going to go from negative to a positive value. So when you follow this on the slide here, we go from the very top, right? There's my resting membrane. My voltage-gated sodiums are closed, and my voltage-gated potassiums are closed. They're right next to each other. Sodium has not yet diffused to this part of the membrane. Here comes my cloud of sodium, making the membrane go from -70 to a slightly more positive 55 value. And you can see that by the slight increase in membrane on your graph right here. As long as we get to at least 55, that opens up the voltage-gated sodium channel. Sodium's going to rush in, because it's following its chemical and electrical gradient. And keep in mind that both sodium channel and potassium channels, these voltage gated ones, they're side by side. They both experience the negative 55 value. But the sodiums are more sensitive. They open up first. And they're going to close first. So for us, to keep this a little more manageable, it's going to be a black and white, 1-step, 2-step, 3-step. No areas of gray at all. No overlapping in functionality, which it really truly is. So I go from negative 55 to a plus 30. And the reason it stops at plus 30 is the doors have slammed shut. You have now closed the voltage-gated sodium channel. There's no more way for sodium to come in. So it can't become more positive.
Why did they close? Is that because of the voltage?
That's because of the stress on the amino acids. They only hold their shape for about half a millisecond.
Okay.
And then they just slam shut. Now, we're going to think of it as this. We open up voltage-gated sodium. Sodium rushes in for about half a millisecond. Those doors close. Now we open up the voltage-gated potassium channels. We open up those puppies. Potassium is going to diffuse out, which allows us to make the membrane now way more negative as potassium leaves. So there's the closure of our voltage-gated sodiums, right? We're now going to open up the voltage-gated potassiums. Potassium gets to diffuse out. Why is potassium diffusing out of the cell?
Concentration gradient.
And it's electrical gradient, right? It's positive on the inside. It helps to repel the potassium ion. And there's more potassium inside than outside. So it's following its chemical and electrical gradients. And you gave it away because you opened up those chemical or those voltage-gated potassium channels. So with the opening of the voltage-gated potassium, potassium diffuses out, making the cell more negative. That's repolarization. All right? Going from a negative to a positive membrane is depolarizing. But when you repolarize it, you're going from positive back to a negative value. And that's the repolarization phase. Now, keep in mind that the voltage-gated potassiums were slow to open, and they're going to be slow to close. They are so slow to close, in fact, that by the time they are done closing, the membrane has gone past its negative 70 value. You are now more negative than you were to begin with. It takes it down to maybe about a negative 80. We now have to go from that negative 80 value back to the resting membrane. And to keep things simple, we're going to say that it's simply due to the sodium-potassium pump. Think about what that pump is doing with a negative 70 environment. Here's your cell. We are at a negative 80 millivolt value. I have a sodium-potassium pump in the membrane. And I know it's going to bring in 2 potassium ions. Won't that help to make that more positive? Right? And the fact that it's also really negative, if any potassium tries to leak out, it's going to try and keep it in too. So the sodium potassium pump helps us to reestablish that resting membrane value of a negative 70. That's the short version of the answer. It's not a UW answer, but it's a short version for us. So I can go from negative 80 with the sodium potassium pump, pumping in more potassium ions, taking us back up to a negative 70, and I'm ready to start all over again. This is one action potential. From the opening of the voltage-gated sodium channels, with the closing of voltage-gated sodium and the opening of the potassiums to repolarize is one entire action potential, and it's happened in just one segment of the membrane. So it's only happened right there. I've got all this part of the membrane to do still, so I'm going to do it over and over again. It's like watching a wave in a stadium. When the arms go up, that tells the next folks that when these arms go down, they're going to start their part of the wave. And so as the arms go down, the next part knows to start their wave. And it's moving around the stadium section by section. Make the stadium an axon. Although our neurons aren't circular. So we're making a big, long, straight stadium, like a horse racing stands. So let's look at the whole thing in terms of its anatomy. There are parts to an action potential. we have the depolarization phase, which is taking us from -55 to a +30. The repolarization is from the +30 back down to, well, -55. And then we have what's called the undershoot, because we went under the -70 value. And then we have our normal resting membrane, which does not experience any stimulus. Right? Remember that the depolarization is due to sodium rushing in. Depolarization is due to sodium rushing in. Repolarization due to potassium leaving. The ions are moving because of their chemical and electrical gradients, and because you've opened up voltage-gated channels for those respective ions. Let's stop there for a second and do a little reality check. It was a little on the fast side. Is everybody okay with the steps of what's happening in an action potential? Keep in mind, we only did that in the initial segment. We can only do this when there is a node region. You cannot do this in an internode, because you have that cell's membrane of a myelinating cell preventing any ions from crossing the membrane. And if ions can't cross the membrane, you cannot change membrane potentials.
So that graph of like the stages of an action potential, that only happens within that node, and that's continuing to happen down the axon?
Right, which is what this whole principle here for this slide is here.
Okay.
All right, so what she just described is what's known then as a saltatory conduction, or a leapfrog effect. Action potentials only happen in the node region. So I'm only making action potentials here in this node. The very last set of voltage-gated sodium channels, if we kind of make this, let's say, an internode, and this space and that space a node, if I am sodium coming in through these voltage-gated sodium channels, and I diffuse this way, there's no channels for me to activate here. So I'm moving along, doing nothing, doing nothing, doing nothing, and I pop out to an exposed node. Well, now there's a whole new set of voltage-gated sodium channels. You can open up them, and a whole new batch of sodium is going to rush in. You can see my effect in the node region, but these very last set of sodium channels, when they open up, sodium rushes in, diffuses this way. I'm back in the tunnel, so to speak. I'm back along this part, and since there are no channels of any kind, there's nothing for me to change. I have no effect until I pop out until the next exposed region. Remember, this is surrounded by extracellular fluid. It's surrounded by high concentrations of sodium, high concentrations of calcium, high concentrations of chlorides, and you're simply just looking for a channel to open up to cause those ions to move if they can move. So it only happens in the node region. That's what's known as saltatory conduction. However, if you have no myelination, then you have a continuous place to create action potentials. Since this neuron has no Schwann cells or no oligodendrocytes, it contains no myelination. That means the entire length of the axon will continuously experience an action potential. These tend to be slower. Unmyelinated neurons send electrical signals much, much slower. Unmyelinated neurons send signals much, underlined, slower compared to a myelinated.
Why is that?
Basically, it's faster for me to skip places to have to, rather than trying to cross the cell membrane every single nanometer by nanometer of region. So here, I'm not being influenced by having to slow down, open up a gate. I'm just buzzing right. It's like the express lanes in a sense. It also saves the neuron energy. Anytime ions cross the cell membrane, you need a pump. I only need pumps where I've got exposed node regions. I don't need pumps along in here, but I need pumps along my entire length, which means more ATP expenditure. So it's even cheaper for energy expenditure.
What's the benefit of having an unmyelinated neuron?
I don't need to maintain Schwann cells or oligodendrocytes. And they will be easier in the sense in terms of I can fit them in places where I want or I need really short neurons. Because you're only going to myelinate long length neurons. And so, relatively speaking, it's more efficient to have an unmyelinated for a short distance and more efficient to have myelinated for long distances. All right, let's make sure you guys understand an action potential. So this will make us realize if you got this or not. Your ability to send the very next action potential is due to what's called a refractory period. Now, there's two parts of a refractory period. There is what's known as the absolute refractory. Focus on what absolute means. What does absolute mean? What does absolute mean, folks?
Definitely.
Definite, final, right? Like my goddaughter teases me and says, "You're going to absolutely reject my first boyfriend." I said, "You're damn right I am. The very first one, I'm going to just take her off the bat." That means no way. All right? Now you've got also a relative refractory period. What does relative mean? And what?
It's all relative, it's not for sure.
Don't use the word in the definition. What does relative mean?
Not definite.
Not definite. What's another way to think about relative? Difficult changes, or maybe it depends, right? So absolute means there's no way you can start a second action potential. No matter how hard you stimulate that neuron, it will never start a second one. If, however, it is experiencing a relative refractory period, there's a chance it could, as long as the stimulus is strong enough. So there's a chance you can make one, but it has to be a strong enough stimulus. Now, why is that true? So let's look at this graph of an action potential and walk you through absolute and relative refractory periods. Over here, I have the point of actual depolarization, the point of actual stimulus to reach threshold. So you've activated your voltage-gated sodium channels. Sodium is rushing in, making the membrane become more positive. Right? That's the depolarization phase of an action potential. But realize that point 3 is halfway through my 1 millisecond worth of time frame. So it's like watching a voltage-gated sodium channel open and then starting the stopwatch and saying, what happens during your first half of a millisecond? And sodium is rushing in. But the second half of that millisecond, as Brandon was asking, the amino acids can no longer hold that position, and they slam shut, and they're unresponsive. The proteins have to redistribute their charge. And during that time, if you apply a stimulus to that same voltage-gated sodium channel from the 0.5 to the 1 millisecond, it's never going to respond. It's sort of like me saying, if you were to reboot your computer, can I use it while it's rebooting? No, you absolutely cannot. You must wait for it to fully reboot. Now, once I let the first millisecond of time elapse, for the second millisecond, is there a chance I could start another action potential? Is there a chance I can open up that same voltage-gated sodium channel? Well, yeah, if the stimulus is strong enough, because now you're in the relative refractory period. This is about time elapsing, time passing for that voltage-gated sodium channel. So if we look at the membrane right here, look at the distance from where threshold, or sorry, where threshold is. It's this dotted line, right? This white line represents -70. So cross that -60 out, make it a -55, so we can be consistent in all of our notes. This is a different neuron they were using. So make us a -55 here, and then realize the difference between -70 and -55. It's a 15 millivolt difference. So let's pretend, just to make things simple, if I'm looking at a graph here, and here's resting, and here's threshold, and then here's my action potential, coming up here, going up, coming down here, down there, and back to this. And if we can look at the distance here between -55 and -70, it's a 15 millivolt difference. Let's say we get 1 millivolt for each sodium ion. It's only going to take 15 sodium ions to go from resting to reach threshold in our fictitious little analogy here. But what if I was right here in time? Is this more negative than -70? Yes. Let's say it's -80. Well, What's the distance now in absolute values between negative 80 and negative 5? It's a 25 millivolt difference. Now I need 25 sodium ions. I need a stronger stimulus to go from here right to there. What if I was right here instead? Is that a little more positive? So I need a little less sodium. Still more than normal though, right? I still need 15 millivolts here. Maybe this is an 18 millivolt difference. But as more time elapses, especially from this part here, I need less and less stimulus to reach threshold. If I wait long enough for the second, right about here, so this is the absolute area here, but from here to about here is a relative refractory period. But the longer I wait, the less stimulus I need. And if I can get to this part, it's a normal resting membrane potential. Then back to this distance. And what you're looking at is how much time has to elapse for that specific voltage-gated sodium channel to be able to respond to another round of stimulus. So let's sort of do it like this. We're going to make this the initial segment. I'm going to have voltage-gated sodium channels to each one of these chairs. We're going to have invisible, pretend voltage-gated potassiums in there. But they're not a part of the story because it's the sodiums that make the membrane more positive anyways. So here comes sodium from a chemical-gated sodium channel. I diffuse to the initial segment. We reach threshold, pop open those voltage-gated sodium channels. Sodium rushes in. Which way is sodium going to diffuse? All directions, right? Those that go this way do us no good at all because these are all chemical-gated channels. But the sodium diffuses over here is going to activate this voltage-gated sodium channel. Now, as the sodium is diffusing this way, I'm finishing my absolute refractory phase, perhaps, or just getting into it. The sodium is going to get here and open up this gate. This sodium that comes in, where is it going to go? Wherever there's less of it, right? It's going to go this way, because there's more here than there is here. I'm going to diffuse over here. But this channel has not finished its absolute refractory phase yet. So this sodium won't make this guy open up. Now, if the sodium comes in here, we'll diffuse over here. I'll open up these sodium channels, but this guy just opened up. But those guys have had more time. So these guys are going to be in their absolute. Sodium that comes in here, will it diffuse over here? Yeah. But the channels right behind it are unresponsive because they're in their absolute. So this sodium has no effect. But what if it diffuses further over here? It will. And these guys will be in their relative. But there's not enough sodium anymore. This is a graded potential going backwards. It gets so weak that it won't make this guy open up. But what if sodium came in from another set of chemical-gated channels? It was a whopping dose. Then yeah, we could start another action potential here. So I'll have one happening here, over there, one happening over here. So what you think about it is, if I were to stop time and look at, say, this channel opening up, if I could freeze time, this channel is open, so sodium is rushing in. These guys have yet to be stimulated, so they're all in their resting membrane. This one's currently experiencing an action potential. The channels right behind it are always in absolute. The ones behind them are always in relative. And those behind them, if there were, would be back in the resting membrane potential values. So in a sense, you're sort of looking at multiple stages of time, where I could be looking at sort of like this idea, maybe, of if this is 3 segments of my membrane in a row, use purple, that the sodium channel currently experiencing an action potential. The ones right behind it are always in absolute. They will never respond. The ones behind them have had 2 milliseconds of time elapse. They're in the relative, they may or may not. The third set are back to resting membranes. And it's always sort of that cookie cutter pattern along the length. Because of that, and because we're starting at the initial segment, when action potentials get created, they're only going to go one direction. They're forced to go from the soma toward the axon terminals. they cannot stop and turn around and head back towards the soma, because the gates right behind them are always an absolute. And they'll have enough oomph to stimulate the ones that are further behind in time that might be in their relative, because there's simply not enough sodium to reach them. And this is what's known as the refractory period. It's also what forces an action potential to travel in one direction, as long as it starts at the initial segment. So what would happen if I had a fictitious neuron, and we got channels here, 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 and here, all voltage-gated sodiums. I apply a voltage right here on a resting neuron. Which way will the signal travel? Why both ways? And both are in the resting membranes, exactly. So you think what's called a voltage patch clamp? It's a cell's version of a roach clip. And put it on the membrane, apply voltage, open up these sodium channels, and as you said, sodium will rush in. But since both sides of the application of stimulus are in the resting membrane, you'll send a voltage action potential toward the soma. When it gets here, it's going to peter out because there are no voltage-gated channels. But it'll diffuse in this way and head this way, all the way to the axon terminal where there's voltage-gated calcium channels. You open up them and you release neurotransmitters. This is how your body sort of, in a sense, prevents the message from going the wrong direction. It's the absolute refractory period that's always behind in time to the currently open voltage-gated sodium channel, which is what I'm trying to portray in this idea in a slide I made for PowerPoint. You're looking at one voltage-gated sodium channel, but how it changes over the course of time. So here you've got the one that's currently open, down toward the axon terminals, they have yet to be stimulated. So once this channel causes sodium to come in and to create the action potential, it's then going to go through an absolute refractory phase. That'll last about overall 1 millisecond of time. Then from 1 millisecond to 2 milliseconds of time duration, it'll be experiencing its relative refractory period where it might be able to, as long as you bop it on the head hard enough to create one. And then if you let more than 2 milliseconds go by, that exact same voltage-gated channel will be now in its resting membrane phase. And all it needs now is a nice normal stimulus. This is the same channel experiencing elapses of time, and how much time has to elapse to go from resting or currently activated to absolute to relative, and relative back to its resting. It's a nice, easy, basically 1 millisecond for each phase. You guys okay with that? Okay. So then, that's how we can have this kind of change in communication from neuron to neuron. If we look at creating a stimulus in the precentric neuron, it's going to be an action potential. You're going to open up voltage-gated sodium, sodium rushes in, potassium channel opens, potassium rushes out. So it's sodium and potassium out, only going in one direction because of the relative refractory period, and most specifically because right behind it is that absolute phase, so the action potential can't turn around behind it. That sends an action potential down the entire length of the axon. It gets the synaptic knob, where we go from an electrical signal to now the release of a neurotransmitter, which is a chemical signal. As we just saw, the neurotransmitter lands on the postsynaptic membrane, opening up chemical-gated sodium channels, causing a graded potential to happen in the soma. Another name for it is synaptic potential, because it's happening in the synapse. It's the same thing. Hopefully that graded potential of sodium will diffuse through the soma, reach the next initial segment in the postsynaptic membrane, enough to trigger a threshold to start it all over again. So if we had two neurons in series, we would go from an electrical signal to chemical language or signal, back to electrical, back to chemical. We're just changing languages. So now we're going to look at how we can screw up the formation of an action potential. So let me give you guys about a 5-minute break. Think about, if you truly understand how an action potential is formed, first of all. And if you don't, let me know as soon as you walk back in so I can get that fixed, because now we're going to screw it up. Figure out ways of making it harder to form. So we'll give you guys a 5-minute break and come back in 5 minutes.
Okay. You left something?
No.
Get it.
She knows what I'm saying no to.
Excuse me, do you have a Band-Aid? Huh? Do you have a Band-Aid?
Two different schools. Time, session, waiting session.
It just changed my wallet.
Oh, that's it. That's okay.
Open it, even more, before the stronger set is good. And you do micro and springtime. But if you don't do 42, springtime.
You don't really tell me one.
Yeah, it's works too, I think. Oh, sorry.
They're from my work.
It is going to be really demanding.
I don't know.
Are you sure? You can use them. I don't know if it works.
My A students were struggling. They did it.
I know.
But I saw them.
Maybe I can ask Saturday. Thank you so much. Maybe I can ask.
Okay.
Yeah, thank you so much.
And then they did 4 hours of micro. And then the next day, did 4 hours of 242, and 4 hours of micro. You have a lot of lab reports to do, a lot of lab procedures. Think long and hard on this. Okay. Now there's a chance. But right now, looking at current skill sets, I want to save you from that part. And I would suggest that don't do it. Now let's see what happens at the end of this quarter. If you make what's possible to do, then that shows that you can put the nose to the grindstone. You have the skill set to be able to do it. But why do you feel like miserable? If you don't need to do it, don't do it. Micro is hard. Are you? So you're going to take and fall. Okay. Her sister told me.
I was like, I don't know.
So that's good, right? But if you're thinking about it, you don't want you to do that. Okay. Of course. You're starting from scratch all over again. It's only half of phys. You have respiration, cardiac, endocrine, digestion, renal, reproductive. Most of your phys comes in the second quarter. You have to do both. Yeah. To get AMP, get your equal equivalent of 231, 232, you have to do all 241 and all 242 to get the translation of those two quarters.
So what I did for the last week's lecture was I went home, I recorded it, so I took a snippet. I haven't really kind of went over that yet, like sat down and studied. Well, just over these past couple of days.
What did you do on Friday?
Did I do Friday? Yeah.
I just want to see what it says.
Well, I didn't do all in one sitting. It took me a couple of times. It's Saturday.
Yeah, 2 counts of, 3 counts of middle abdomen, or 2 counts of,
Yeah.
Alright, that's your like crash thing. But that's good. You'll start because you know what, those are follow them and powerful. Yeah. The bottom, either on the first line, and go to the next line. Don't go one, two, three. Step, and the footing, this all footing, and then the next step. But until that footing is firm and understanding, don't think of a step, it's all like.
So also last time you mentioned the oligodendrocytes and the Schwann cells was that one can repair and one can't. Is this oligodendrocytes?
Schwann cells repair for,
Right, well, right, when you do a marker, you're gonna have zero in the middle, down about, middle of the wire. You're gonna have some bars going down, some bars going. All right, so let's have you guys rally the troops for a second. Is everybody okay with the sequence of the events that are needed to start an action potential at the initial segment? Is anybody confused about, I do not understand why this is happening or what this part is about? You're all okay with the idea of a refractory period, having both an absolute phase and a relative phase? And why these can only go in one direction? You guys are okay with that? I guess an absence of comment means you're all okay with it. All right, so we're going to move on. We actually would like a response from you guys. Like, yes, we did it, and we don't, we need more time. Exactly. Otherwise, you think, are they even listening to me? You guys even alive out there? All right, so since nobody said anything, I'm going to assume that means you have at least, you think you understand it, you just need more time to make sure. Now we're going to look at how we can influence a neuron's ability to actually create an action potential and 3 different mechanisms. One is going to be influence the membrane potential at the initial segment. So we're going to screw around with this. Then we're going to influence the release of neurotransmitters from over here or over there. Then we're going to influence the duration of the neurotransmitters at the synaptic area. So here's the first one. Get your bearings, look at the diagram. What is the diagram showing me? I see a big green synaptic knob making a synapse with maybe a dendrite or a soma portion of a neuron. There's its initial segment, which means that's the axon. Here's a graph of my membrane potential over time. There's my threshold value. So we're looking at neurotransmitters can excite postsynaptic neurons. So this is the postsynaptic, that's the presynaptic. If this synaptic knob were to release acetylcholine, acetylcholine is known as that excitatory neurotransmitter. When the green synaptic knob releases acetylcholine, it'll land on chemical-gated sodium channels, right? And that will cause sodium to diffuse into the dendrite, create a cloud of sodium ions, a graded potential, and we can see the effect. It makes the membrane slightly more positive. And those sodium ions will diffuse to where there's less of them, and they're going to diffuse to the initial segment area. But realize they're not reaching threshold. They're just making it more positive. So if another stimulus were to come behind it, you might actually reach threshold. The purpose of this is to excite the neuron, to bring it close to threshold, so if a stimulus were to be applied that would normally not be able to trigger a response, you will actually now create one. Your real-world scenario is sort of like the notion of heightened senses or heightened awareness when you're scared. That means you're releasing excitatory neurotransmitters, you're activating lots of neurons, but just bringing them closer to threshold, but not actually at threshold. Which means it's easier for you to hear the sound, smell the smell, or to see the thing moving. So it takes a weaker stimulus to actually activate it. Now your senses are heightened. And that's the whole purpose of acetylcholine in this was called EPSP. Right? Down here in the lower right-hand corner. Excitatory, because we're trying to make the membrane slightly more positive on the postsynaptic membrane. What's known as an EPSP. It's using acetylcholine and it is slightly depolarizing the membrane. And look what happens to the membrane potential as time passes by. It gets weaker and weaker and weaker. That is your graded potential. Unless you apply another stimulus right away, you lose the benefit of making this membrane slightly more positive. Now, if that will make sense, then we can think about doing the exact opposite to that initial segment to make it now more negative rather than a slightly more positive. In which case, now we're looking at an inhibitory response. How can I make the initial segment more negative compared to usual? Well, we still have a presynaptic neuron in red, a postsynaptic structure in beige. This is what's known as an inhibitory postsynaptic potential, abbreviated IPSPs. Well, how can we do this? Well, now we have an option. In the first one, you guys were using acetylcholine, which you recognize as being used for skeletal muscle contractions, but also as an excitatory neurotransmitter as well. Still landing on chemical-gated sodium channels. But now you have a new neurotransmitter to think about, and the abbreviation is GABA. When you write GABA, it should always be in all capital letters. You do not need to write out the full name of GABA aminobutyric acid. Yuck. GABA is much faster and much simpler. It's a neurotransmitter released from my red presynaptic knob. But here's the neat thing about GABA. It can land on one of two different possible chemical-gated channels. It could bind with a chemical-gated potassium channel, in which case, if it does, right, it allows potassium to diffuse out of the cell, right, because there's more potassium in than out. So as potassium leaves this area where those chemical-gated channels are, that makes this area, relatively speaking, compared to a moment ago, more negative than usual. Well, that more negativeness will attract anything positive, even positive things away from the initial segment. And as those positive things leave, it, relatively speaking, makes the membrane here now become more negative. And we can see that with our graph. There's the membrane becoming more and more negative. It was simply due to having potassium leave, which makes this region slightly more negative than usual, and it'll attract any positive thing to that more negative area. And if those positive things happen to come from the initial segment, then we have a hyperpolarizing effect. We are now making it difficult, more difficult for this neuron to reach threshold at its initial segment. Does the logic behind that work for the moment? All right, question now is, when would you do this? When would you want to actually make it more difficult to inhibit a neuron from firing? When you guys are sleeping. How many of you have dreamt and you're dreaming about running or skydiving or flying? Are you running in bed while you're dreaming about running? No, you're not, right? Why? Because your brain is releasing a crap load of GABA onto all your motor neurons in your brain. You are inhibiting them from actually acting out what you're dreaming about. But now, how many of you have watched your pooches and they kind of bark and twitch when they're sleeping, right? That's a lapse in GABA application. Or when you're sleepwalking, GABA's not working there. And so you're actually acting out what the brain is trying to make you do. Well, why would you not want to act out your running, your jogging, your dancing, your hunting skills while you're asleep? How would you hurt yourself? Not jump out of a window. Think old school. Think evolution. Where did you used to sleep hundreds of thousands and millions of years ago? In the branches of trees. If you fell off a branch, you A, might break a limb, or who might you land on top of? Oh, a lion, a tiger, a saber-tooth. Not a good idea. So your brain says, all right, we're going to paralyze you while you're sleeping. Here's your really cool evidence of this. How many of you have been studying physiology really diligently late at night or on a Saturday afternoon, and you're on the couch and you kind of fall, maybe you jerk yourself awake because you have the sensation of falling, right? And your arms go out. That's your brain saying, dude, you're falling. Grab a branch, all right? Because you've only been sleeping on beds for a couple hundred years. compared to millions of years of evolutionary programming to sleeping on branches, right? That's why you can take a baby, like a 2-month-old, and just toss them in the pool. They're going to do a parachute pose, because they're trying to grab branches, they're going to hold their breath, and they will not drown. It's pretty cool. That's okay. That's not an experiment.
It's for science.
It is. It's outrageous acts of science. Okay? So now what if we just change the ions? What if instead of having potassium leave, you instead opened up chemical gated chloride channels? First of all, which way would chloride diffuse? It's going to go into the soma, creating a relative cloud of negativeness, like potassium would if it were leaving. So now, even though they're not showing it to you, you can imagine chlorides coming in, creating a negative cloud, which would again trap more positive things away from the initial segment. Or maybe chlorides would diffuse and get here. Either way, you're making the membrane more negative. It's also a very short period of time. If you do not act upon it, as the milliseconds progress, you're going to go back to normal, and back to your normal resting membrane value. So we have one way of doing that excitatory post-synaptic potential, the use of acetylcholine onto chemical-gated channels. The whole purpose is to make the membrane slightly more positive, but not to reach threshold. I want to stimulate the neuron, not actually create an active potential. Or I can do an inhibitory fashion, through an inhibitory and focus on postsynaptic structure. I can use GABA to land on chemical-gated potassiums and or chemical-gated chlorides. Either way, it results in the initial segment of my postsynaptic neuron becoming slightly more negative, so I make it now more difficult for this neuron to be able to form an action potential. Really focus on the words postsynaptic, because we're going to do something different in a moment. All right, now let's look at other ways of either inhibiting or exciting the initial segment in terms of the pattern of stimulus. We have temporal summation and we have spatial summation. Look at the picture and look at the difference. In temporal summation, I have one synaptic knob that's going to be repeatedly stimulating the neuron. And they're trying to show that to you with these changes in grades of reddish and pink. Here's one stimulus. See how it's getting weaker the further it travels? Like sodium does, like a graded potential. But if I can summate another stimulus right behind it, build upon it, I get a stronger stimulus overall. And maybe it'll reach the initial segment, and I can either inhibit or stimulate this neuron. So one way to think about it, temporal summation is a single synaptic knob that's going to be rapidly stimulating the postsynaptic neuron. A single synaptic knob that's going to be rapidly stimulating a postsynaptic neuron. Keywords being single and rapidly. Let's contrast that now with spatial summation. Now we're looking at two synaptic knobs, but they both must be stimulating at the same time. So it's two or more synaptic knobs simultaneously, or at the same time, whatever word you want to use, at the same time stimulating the postsynaptic neuron. Two or more synaptic knobs stimulating the postsynaptic at the same time. If those key words are not in there, then this question on page 3, worth 4 points, is going to cost you points. So I'm going to ask you this, I promise you. Which reminds me, my intellectual fantasy of, the 4 point, it bombed. Didn't get it yet again. I'm trying with this question, okay? I'm going to ask you this. How do you define temporal summation and spatial summation? Key words: single synaptic knob, rapidly stimulating, two or more synaptic knobs at the same time. And the picture shows you that, hopefully. All right, now let's look at influencing the release of neurotransmitters. This is why I said to focus on the previous conversation of postsynaptic potentials. We're now going to screw with your mind a little bit. Your normal synapses from neuron to neuron normally are axon terminals to dendrites or axon terminals to somas. Those are your normal synapses. There is a case where you have the rare event of the axon terminals of one neuron synapse with the axon terminals of another neuron. Okay, so it's axon to axon here. That's what this is trying to show you. All right, the purple thing is my traditional postsynaptic neuron. The gold, whoops, wrong direction. The gold is my traditional normal presynaptic neuron. And the green dude is trying to influence the presynaptic neuron. That's why this is called presynaptic inhibition. This is not post. We are influencing the gold neuron's ability to release its neurotransmitters. So how do we do this? We take the green one and talk about an action potential coming down its membrane, opening up voltage-gated calcium channels. Calcium is going to come in, and now this green synaptic knob is going to release GABA. GABA is going to influence, and here's the weird thing, GABA is going to influence voltage-gated calcium channels. Yes, GABA is a chemical. This is where the bizarre thing is happening. It's actually influencing voltage-gated calcium channels and preventing them from opening. Now that will make sense. If this GABA is influencing or inactivating these voltage-gated calcium channels, then when the normal action potential comes down, my gold presynaptic neuron, and it reaches those voltage-gated calcium channels, there's going to be fewer of them opening, which means less calcium is going to come in. Remember, you need calcium to do exocytosis. So if there's less calcium coming in, there's going to be less release of neurotransmitters. And if I have less release of neurotransmitters, my purple postsynaptic dude over here is going to get a less response. That's why it's known as presynaptic inhibition. Now keep in mind that this GABA over here was involved in IPSPs. Now I've got GABA also being used in a presynaptic inhibition fashion. Whereas that GABA was influencing voltage-gated calcium channels, not chemical-gated. This one was influencing membrane potentials at the initial segment. This one's influencing the release of neurotransmitters from a presynaptic neuron. And that's quite a mouthful. We've got to get our heads wrapped around that. And the pictures are trying to show you all of that. All right, we have, now, if you understand this makes sense, then let's look at what happens with a presynaptic facilitation, doing just the opposite. We have now a different neuron making a synapse with the axon terminal of our presynaptic neuron. So the green dude has an action potential, voltage-gated calcium channels open up, calcium comes in. Now instead of releasing GABA, this is a different neuron, it's releasing serotonin, which is also going to influence the ability of these voltage-gated calcium channels. They're now making it easier to open up. In which case then, when for my gold presynaptic neuron, when the AP comes along, it's even easier to open up these voltage-gated calciums. That means more calcium comes in, I get more neurotransmitter release, and whatever my postsynaptic purple thing is, it gets a bigger stimulus. So that means I've given you acetylcholine as an excitatory neurotransmitter, GABA as an inhibitory neurotransmitter, or inhibitory postsynaptic mechanisms, But that same neurotransmitter coming from a different neuron on a different application can also be involved in presynaptic inhibition. And then we have serotonin in presynaptic facilitation, making it easier for the purple dude to become activated or to cause the gold or yellow presynaptic neuron to release even more neurotransmitters. So, in a nutshell, what this sort of is if I want to make something slow down, I can use a presynaptic inhibition pathway if I want to slow it down. If I want to magnify it, I could do a presynaptic inhibition. I'm sorry, facilitation, pardon me. A presynaptic facilitation. Well, first let you get used to these before I talk about, well, what if we did this on next Wednesday? But you can apply these also on top of EPSPs and IPSPs as a way of magnifying those functions. And we'll address those on Wednesday. So my suggestion is you make maybe a list of IPSPs, EPSPs, presynaptic facilitation, and presynaptic inhibition. List the neurotransmitters and the channels involved. You kind of used to thinking about the different processes in which neurotransmitters they use and what kind of a gated channel are being activated or used in that mechanism. You've got 4 different ways to keep things straight. Now let's look at influencing the release of the neurotransmitters, how long they actually get to stay in the synaptic cleft. So we have some more definitions for you, because you're going to come across these quite often in other biology classes. Remember that we can organize neurons based upon their structure, right? You've got anaxonic, unipolar, multipolar, bipolar. I can label neurons based upon their function, right? Sensory, motor, or interneurons. I can also classify them based upon the type of neurotransmitter they release. If a neuron releases acetylcholine, it belongs to a cholinergic category. I'll write it up here. It's known as a cholinergic neuron. If, however, my neuron instead releases norepinephrine or epinephrine, it's known as an adrenergic neuron. So I've got a cholinergic family of neurons or an adrenergic family of neurons, depending upon what neurotransmitter they release. Now, if they're releasing acetylcholine, then there's going to be a family of receptors known as cholinergic receptors, because acetylcholine is a cholinergic neurotransmitter. So I need catchers for my cholinergic neurotransmitter. The 2 specific types of cholinergics, one are called nicotinics, the other ones are called muscarinics. These are specific types of cholinergic receptors, and they're found in different parts of the body. The nicotinics you guys have in your motor implants. Remember, those were with skeletal muscles. So nicotinics are found on skeletal muscle tissue. When you get into 242 and do cardiac, you're going to find the muscarinics are on cardiac muscle cells. They're not on skeletal. These are very tissue-specific locations. So nicotinic for skeletal muscle, muscarinic for cardiac. It's the same neurotransmitter, but it needs different receptors for different tissues. Same is true for norepinephrine. If you're releasing norepinephrine, you're in an adrenergic neuron, and we have an adrenergic family of receptors. The 2 specific types of adrenergic receptors are your alpha family and your beta family of receptors. All right? Alpha and betas. Now, we have a couple of different kinds of alphas and betas. They've actually discovered some new ones. We're not going to get into every single where do you find B1s and B2s and A1s and A2s. But for right now, the nice rule of thumb is that bless you, if you end in a 1, so if you're an alpha 1 or a beta 1, you tend to be excitatory. So alpha 1s and beta 1s are excitatory. And if you're an alpha 2 or a beta 2, you're inhibitory. The general rule of thumb. Little shorthand note, if you're trying to abbreviate, don't do beta as that. That's a B. This is a beta. It's a tail. Okay, it's like little grammar for shorthand. All right, so alphas and betas are two specific types of norepinephrine or adrenergic receptors. The alpha 1s and beta 1s are excitatory. The beta 2s and alpha 2s tend to be rather inhibitory. Now, let's put them in use and also learn a little more about our sympathetic nervous system. We're looking at just the middle part of our graph. We're looking at the autonomic nervous system and what happens in the sympathetic versus the parasympathetic. Here's my first neuron, which you guys would think of as your presynaptic, and then there's my synapse, and there's my postsynaptic. In this scenario, under the sympathetic, you have short myelinated pre-neurons, long unmyelinated posts. Short myelinated pre's, long unmyelinated posts. Now, the pre's, otherwise more known as preganglionic neurons, they're going to release acetylcholine onto the postganglionic neuron. And since you know that they're releasing acetylcholine, then you know they have to have some kind of a cholinergic receptor for them. And when the postganglionic neuron is activated, it's going to release norepinephrine, not acetylcholine. That norepinephrine could land on one of these major types of target tissues. Smooth muscle tissue, like around blood vessels, could land on glands, or could land on your heart. Whether it is either one of these, we don't care, but you realize that these now have to be adrenergic receptors, no longer cholinergics. And for right now, for the heart, you can think about beta 1. Put a beta 1 next to the heart. So for my sympathetic nervous system, the first neuron, the preganglionic, I know is short in length, is heavily myelinated, will release acetylcholine onto the second or postganglionic neuron. That means that the dendrites and soma have to have some kind of a cholinergic receptor that is going to release norepinephrine from my postganglionic neuron and land on some type of major target tissue. Maybe it's blood vessels, glands, or heart. And for the heart, since it's norepinephrine, it will be specifically beta 1 adrenergic receptors. And in 242, you will learn that cause increase in heart rate and force of a contraction. Now, the parasympathetic is in a way kind of the opposite. If the sympathetic was short and myelinated, then the para, the very first one, is actually long and myelinated. And its post is short and unmyelinated. Now the nice thing is it uses acetylcholine throughout its entire system. The preganglionic neuron releases acetylcholine, which means then that the dendrites and soma of the post have to have some kind of a cholinergic receptor. That post neuron will release acetylcholine onto some type of target tissue. And if it happens to be the heart, it's going to be muscarinic receptors. When you look at this, realize that we're using acetylcholine in all of the parasympathetic and the first part of this bless you sympathetic. The only oddball is this long, unmyelinated, postganglionic neuron of my sympathetic division. It's the only one not following suit. Otherwise, everybody else is acetylcholine. Everybody else has to be a cholinergic between these two. The middle one, you'll get more in endocrinology because it's an odd case where you have just a single neuron releasing a neurotransmitter that causes a hormone to come out of its target gland. They're not two neurons in sequence. So I'm not going to focus on this one. You'll learn about that one in 242. We're focusing on this upper branch of sympathetic, right? The lower branch of parasympathetic. Recognize which ones are myelinated, which ones are short, who's releasing what neurotransmitter, and what receptors they have to have, and then what are their possible targets, and what kind of receptors they would need to have for those. Good flashcard material. And then the last little bit. Now let's influence the duration of the neurotransmitters that are found in our synaptic cleft. We can end the effects of the neurotransmitters by degrading or breaking down the neurotransmitter, in which case you'll have a very specific enzyme for that neurotransmitter. Most of those enzymes are found in the membrane of the postsynaptic structure. Most of the enzymes are found embedded in the membrane of the postsynaptic structure. They're already there waiting for the neurotransmitter to show up. Well, if we're using acetylcholine, then the name of the enzyme is acetylcholine esterase. Remember, you can recognize an enzyme because it ends in ASE. That's your hint that it's an enzyme. And they tend to be named for the neurotransmitter they target. So the whole function is that I need to break apart my acetylcholine into two parts. And since you broke the neurotransmitter, it can't work anymore. Therefore, it cannot land on a chemical-gated sodium channel. You've essentially ended the message because you broke the messenger. That's how you can terminate the signal. You can also have what's called a reuptake process. There are these really cool proteins in the presynaptic membrane. They're sort of like a Ferris wheel. And they literally bring up either the neurotransmitter itself or the parts of neurotransmitters. But if you remove the neurotransmitter from the synapse, then it's no longer there to act as a messenger. You have ended its ability to send a signal. That's called reuptake. Like a giant Hoover vacuum cleaner in a sense, just sucking them right back into the synaptic knob. So I have enzymes to degrade the neurotransmitter, or a reuptake process, which is a little protein structure that brings the neurotransmitter back from where it came from.
And the enzymes are found most of them where again?
In this postsynaptic membrane. Now, if that ends it, let's do just the opposite, to prolong the effects. I gotta come back here for a second. Now, there are enzymes known as MAOs, which stands for monoamine oxidase. That's the name of this enzyme, which means any neurotransmitter that has a single amino acid in its structure will be broken down by an MAO. Things like norepinephrine, dopamine. They can be affected by MAOs. That's the enzyme your body produces. And it's trying to break apart that neurotransmitter. But if my doctor wants to prolong the exposure of the neurotransmitter, he's going to give me an MAOI. Right? So your cells make the MAOs. Pharmacology makes MAOIs. That's a monoamine oxidase inhibitor. It's to inhibit the MAO from working. If you inhibit the enzyme, it cannot break apart the neurotransmitter. That means it stays there longer and a better chance of binding to its possible receptor. A better chance of activating that postsynaptic structure, whatever it is. Now, next time you guys stay up late and taking your 10-minute break from studying, listen to these commercials that come on late on at night. And they mention MAOIs. If you're on MAOIs, you go, oh, that's an inhibitor of the monoamine oxidase enzyme, which means we must be targeting dopamine or norepinephrine, and the doctor is simply trying to keep that neurotransmitter in existence for a longer period of time, give it a better chance of working. At the same time, we have reuptake processes. We can also inhibit the reuptake process. But there are two ways to inhibit the reuptake process. If you do TCAs, tricyclics, they're kind of a broad spectrum shotgun approach. They tend to affect multiple neurotransmitters from being reuptake. And think about that then. Multiple neurotransmitters means multiple symptoms. You're going to get multiple side effects. And oftentimes, we don't want you to have several side effects, so we need to more specifically target the enzyme, or I'm sorry, the neurotransmitter we actually want to work on. You might take what's called an SSRI, which is a serotonin selective reuptake inhibitor. You're only preventing the reuptake of serotonin. You're not messing around with dopamine or norepinephrine. It happens to be maybe in the area, which means fewer side effects, a more specific effect of the pharmacological drug. So we have two ways of keeping a neurotransmitter in existence. I can provide a drug that inhibits the enzyme from trying to degrade the neurotransmitter, or I can screw around with the uptake protein that's trying to actually bring it up by inhibiting its functioning. Normally, your body's trying to end the activity of a neurotransmitter by having specific enzymes in the membrane to break apart the neurotransmitter so it won't work. But we can intervene with that by giving you inhibitors. We also have reuptake processes that normally try to bring the neurotransmitter back in, which would end the message. But again, we can intervene with that by giving a reuptake inhibitor of some kind. And there are specific families of these drugs for specific neurotransmitters in the brain or the body. Oh, my throat's tired. We'll wait for this one. All right, but it's yet another way of classifying your neurons. Skippy. All right, so we'll be getting with this on Wednesday. You'll be getting a new set of notes because it won't take me the entire lecture to work through the functions of the brain. So I'll send you guys an email when there's a new set of lecture notes to come up for Wednesday. So those who have lab, we'll see you in about 10 minutes. The lab you guys had tonight, we'll be doing on Wednesday. Yes, you have to do it tonight, tomorrow, turn it in on Wednesday. So I think great to get back to you by Monday. All right, lab folks, give me 10 minutes.
No, wait, I have to
Yes, don't skip it.
I want to.
That's